Hey everyone, this is Dr. Medicine RX, and in today's video we'll be discussing about syphilis. So I have divided this topic into various videos as this is a pretty large topic and needs to be covered pretty thoroughly, and this marks the beginning of a venereology series. I will also try to start leprosy simultaneously, and I'd also like to apologize for not posting in a long time because I was preoccupied with work. So coming to the causative organism, it is Treponema pallidum, which belongs to the order of Spirochaetales and the family Spirochaetaceae. In this family, we have a couple of important genera, of which Treponema is one. Coming to the structure, it is a slender thread-like organism which is flexible and motile, and it is non-encapsulated, and it has about 8 to 24 coils, and it has tapering ends. Coming to various measurements, the length of the organism, it varies from 6 to 20 micrometers. The diameter, it varies from 0.10 to 0.18 micrometers. The coil length is 1.1 to 1.1 micrometers, whereas the amplitude, it ranges from 0.2 to 0.3 micrometers. So it is a distance from the center to the edge of the outer edge of the organism. Coming to the basic three important parts of its structure, these include the central protoplasmic cylinder, the second is the periplasmic flagellae or the endoflagellae, and the third is the multi-layered cell envelope. So this central protoplasmic cylinder, it runs the entire length of the organism, and it has nucleoid and ribosomes and other important organelles, and it is bound by cytoplasmic membrane. So the inner cytoplasmic membrane of the cell envelope bounds this, is surrounding the protoplasm, and this protoplasm also has 6 to 8 cytoplasmic fibrils which run along the inner side of the cell membrane. Coming to the periplasmic space, it lies outside the inner cell membrane. That is, it is between the outer membrane of the cell envelope and the inner cytoplasmic membrane. So this is the periplasmic space. So the peptidoglycan layer is present in this periplasmic space, and there are also periplasmic flagellae which are also known as endoflagellae, and because of this periplasmic location of the flagellae, the treponeme is able to move in viscous fluid. So these endoflagellae are again 6 to 8 in number, and usually 3 flagellae are present on both ends. Coming to the cell envelope, we have the outer membrane, the middle thin peptidoglycan layer, and the inner cytoplasmic membrane. So in the sketch below, so the purple colored is the outer membrane, the black colored ring is the peptidoglycan layer, and the blue colored ring is the inner cell membrane. So all these three, they are together called as cell envelope, and between the outer membrane and the inner membrane containing the peptidoglycan layer is the periplasmic space, and the tiny circles are the endoflagellae, and the bluish center is the protoplasm. It has nucleoid and ribosomes. Coming to visualization of the organism, this organism is gram negative, but on prolonged Giemsa staining it appears pale pink. But to visualize the unstained organism, dark field microscopy is the best. And if we want to visualize it under microscope, then silver impregnation technique is required. Coming to the various types of movements shown by this organism, these include slow forward and backward movement, compression expansion movement, bending movement, angling movement, burrowing movement, and our favorite corkscrew-like movement, which is characteristic, then looping, buckling, and undulation movements rarely. Some other features of this organisms are that it is microaerophilic, it is fragile, and so it is easily destroyed on drying or heating. It multiplies by transverse binary fission, and the generation time required for this is 30 hours. This organism can't be cultivated because it requires host molecules for survival. Thank you. Subscribe for more.